Hello. In our previous segment, we have discussed about the interphase. If you can recall, we have already discussed about interphase, where the cell actually prepares itself for the division, Now we start the segment with proper cell division. Now the cell division starts. Actually, we are going to discuss today about mitosis. What is mitosis? Mitosis derived from two Latin words. One is mitos, which means thread. Mitos means thread. This thread is nothing but the spindle fiber. The thread is nothing but the spindle fiber, which actually occurs during mitosis type of cell division. This term mitosis coined by Fleming. This term mitosis was coined by the scientist Fleming, who showed the process of mitosis in animal cell. Today we are going to discuss about different stages of mitosis. Let's come to the point, occurrence of mitosis. Where does mitosis actually occurs? We know mitosis generally occurs in somatic cells. Which may be diploid or haploid. Mitosis occurs generally in somatic cells. But in addition to the somatic cell, in germ cells, mitosis can also occur in germ cells. During gametogenesis, we know, that is the formation of gamete, either spermatogenesis or oogenesis. During gametogenesis, three stages actually occur. One is multiplication, another is growth, and third one is division. That is, first one is multiplication, the next one is division. Next one is growth, multiplication, then growth, and third one is division. Is this during multiplication phase of gametogenesis? Actually, mitosis type of cell division can observed. So, if we say the occurrence of mitotic cell division, we may find that normally it occurs in somatic cells, but during multiplication of germ cells, that is during gametogenesis, in the multiplication phase, mitosis also occurs. Now the stages of mitosis. What are the stages of mitosis? We have already discussed the stages of mitosis during our previous segment. That is, there are basically two stages. One is karyokinesis. Another one is cytokinesis. Karyokinesis, as you know, is the division of nucleus, and cytokinesis is the division of cytoplasm. The karyokinesis again divided into four substages. A. Prophase. B. Metaphase. C. Anaphase. And D. Telophase. So, during the process of karyokinesis during the process of nuclear division, two substages can be seen. One is prophase, next one is metaphase, third one is anaphase, and fourth one is telophase. And after the division of nucleus, the division of cytoplasm occurs. And division of cytoplasm actually known as cytokinesis, which leads to the formation of two daughter cells. Like something like that. This is the mother cells or the parent cell. From that, two daughter cells originates after the division of cytoplasm. These daughter cells possess the equal number of chromosomes than that of the mother cell. The equal number or diploid number of chromosome than that of the mother cell. That means after following the mitosis cell division, the newly formed daughter cell possess the equal number of chromosome than that of their mother cells. So this is the reason why mitosis is also known as equational division. Mitosis is also known as equational division because the number of chromosome remain same in case of daughter cell than that of mother cells. That is the reason why we can say that the mitosis is also known as equational division. So before we go to the process of mitosis proper, let us draw the stage of cell during interphase that we have discussed earlier. This may be the stage of cell during interphase. What will happen actually? This is the nucleus. Since we are more concerned about the nucleus and cytoplasm, so this is the nucleus. This is the nuclear membrane. This is the nuclear membrane, and this portion is the nucleopore. And inside the nucleus, chromatin fibers are found. These are the chromatin fibers. So these are the chromatin fibers. So this is the interphase where the cell is ready for going divisional phase. Interphase cell. So this is all about the interphase cell where the cell is ready for division. Now we start to the next phase or the mitotic phase and the first phase of mitosis, which is known as the prophase. For our convenience, we can divide prophase into two substages. One is early prophase, another is late prophase. Though there is no actual demarcation between these two, as I have said, this is for our convenience. We have divided it into two sub-phases. What actually happened during this prophase stage? To draw the cell like this, what actually happened? Number one, nuclear membrane that you have seen during interphase stage, you can shortly written as NM, nuclear membranes starts to dissociate or disappear. Nuclear membrane start to dissociate or disappear. It will be the nuclear membrane will something like that start to dissociate or disappear. Not very prominent as we have found during interphase stage. Number two, what will happen? In interphase stage, we found centrosome. Here is centrosome. But during prophase stage, the centriole starts to migrate towards two pole. During prophase, centriole starts to migrate towards the opposite pole during early prophase. So the next feature will be centriole start migrating. Centriole starts migrating towards pole. This is the next feature of prophase. Number three, chromatin fibers start condensing. So number three is chromatin fiber, as we have seen here, the thread-like structures in the chromatin fibers. These chromatin fibers condense and form chromosome. It will be something like that. The appearance will be something like that. The chromosome structure is this. So, chromatin fibers condense to form chromosomes. This will. And what will happen next? From the centriole, we see from the centriole what actually happened. Two types of fibers arising from the centrioles. Two types of fibers. Fibers arise. Fibers are two types. One is known as the small fiber. Another is bigger one. The smaller one is known as aster. That's why the formation of aster fibers is actually occur in animal mitosis, since centriole is found in case of animal cell only. And the bigger fibers which actually running from one end to other is known as spindle fiber. These bigger fibers actually known as spindle fiber. So two types of fibers arise from centrioles. One is smaller fiber, another is bigger fiber. Smaller fiber are aster fiber and the bigger one is spindle fiber. This spindle fiber, once more, this spindle fiber are of two types. One is continuous, another is discontinuous. In our next drawing, we can see properly what actually continuous and discontinuous fiber means. Let us draw clearly. Here is the cell. In the prophase, late prophase, what will happen? Two types of fibers actually. These are the smaller one and these are the larger one. And the late prophase, we won't get the trace of nuclear membrane or something like that. Chromosomes are arranged here and there like this. These are the chromosomes. So these are the chromosomes. Larger fibers also start originating. You see these larger fibers. These larger fibers actually known as the spindle fiber. Two types of fiber, you can say. One type of fiber. This is one type of fiber which is not associated with the chromosome. This is known as continuous fiber. And another fiber which actually joins the chromosome, this is known as discontinuous fiber. One is continuous fiber and another is discontinuous fiber. Discontinuous fiber is also known as chromosomal fiber. So at the late prophase stage, we find the structure of the cell something like that. So in the first two stages of cell division, one is prophase, the next one is the metaphase stage. So this is all about prophase stage up to this, and now the metaphase stage occurs. What is actually the main features of metaphase stage? What is actually the main features of metaphase stage? Chromosomes are arranged at the equatorial plane. This is the equatorial plane. chromosomes are arranged at the equatorial plane. This is the main features of metaphase stage. How these chromosomes are arranged at the equatorial plane? By this thing. Continuous as discontinuous fiber. This is the equatorial plane. Equatorial plate or equatorial plane. Chromosomes are arranged at the equatorial plane. So, actually what happened? The chromosomes, if we draw the chromosome, these are the sister chromatids and this is the centromere, sister chromatids, and the spindle fiber attaches at this region. This is the kinetochore region. Actually what happened? The chromosomes are arranged at the equatorial plane by the differential pulling of spindle fiber. That is the differential constriction of spindle fiber. This spindle fiber actually, this part of the spindle fiber pulls the chromosome below, but this part of the spindle fiber will pull the chromosome up. This will pull it below, this will pull it up. Through this differential contraction of the spindle fibers, the chromosomes are arranged at the equatorial plane. So, and during metaphase stage, the chromosomes are most properly or very good visible. The visibility of the chromosome is much better at the metaphase stage. So, in our first two stages, we found the arrangement of chromosomes like this. So, we can say this is the sister chromatids. You can draw it like that because sister chromatids are also there. In our next phase of cell division, we'll find how the sister chromatids detaches from one another and go towards the centriole. So that will be discussed in our next phase. For testing your understanding of this lesson and more videos, log on to www.tubelessons.net.